Hey everyone, welcome back to another episode in genetics. Today we're going to start over. With a completely new direction of what we're going to talk about in genetics. Usually, like the first half of the class is mainly focused on transmission genetics, but today we're going to talk about, and we're going to start talking about, is how DNA replication occurs, especially in chapter 6. Okay. But to answer this question, we need to sort of begin way back in the beginning, where these three different parties, they've analyzed and they wanted to answer the question: is how do we know where our genetic information comes from? So they've already known that DNA is sort of in the cell, and it's sort of just bundled up in there, but they weren't really sure what the purpose of DNA was from the beginning. And a lot of hypotheses came about stating that proteins in these cells are actually responsible for passing along the genetic information of from one individual to another individual. And these three people were the ones responsible for figuring out that DNA contains the genetic information that we need, especially nucleic acids. Okay. So to begin with, we're going to start off with this guy called Frederick Grffith. So this guy, he mainly studied a bacteria. It doesn't matter what kind of a bacteria it was, but he mainly studied a bacteria that had an S strain bacteria and a R strain bacteria. So all a strain is, it's basically a bacteria that just has a slightly different phenotype, but it's the same species. I can't remember what species this was. I think it was pneumococcus. But anyway, so this S strain right here, this bacteria that had the S strain phenotype, this is also called the smooth bacteria. These guys had the ability to kill an organism, so they can kill an organism. But this R strain actually was referred to as R because it was rough in shape, the colony. And then this strain actually did not have the ability to kill an organism. So did not have ability to kill organism. So essentially what he did over here, he had 4 different experimental groups that he wanted to look at. The first 2 was basically use the S strain, inject it into mice, and then the mice would obviously end up dying. Right, that was his first experiment that he did. His second experiment that he did was he took the R strain bacteria, he injected it into the mice, and after injection, they all ended up surviving. Okay. Next, what he did was, so the first 2 experiments, we've already sort of, that's what we've expected to see out of the information that we have so far. But the third one, what he did was, he took the S strain. He first boiled them to kill them, so boiled, which basically means that whenever you're boiling, you're basically killing all the bacteria. And then he added these back into the mice, and then they all ended up surviving. Which makes sense, because if they're all dead, they can't actually provide they're all dead, But then the last one, which was really interesting, what he did was he essentially took those S strain bacteria. He first boiled them, and then in that same exact liquid, he added, after boiling, added the R strain bacteria. And then he added this whole mixture back into the mice. And what happened was that these mice, they ended up dying. So what happened here was that this individual Grffith, what he did was he took the mice, he took their samples, and he grew them out on an agar plate to see what happened to these R strain bacteria. And what happened was that these R strain bacteria all ended up looking like a smooth strain bacteria. So basically, what happened here was that the R strain basically transformed phenotypes into the S strain. Right, so this is basically what he found out. The main key takeaway, especially from a genetic standpoint, because he wasn't really focused on answering our question here, he just sort of stumbled upon it. But the thing was that essentially something from this S strain over here, something from this S strain is basically allowing us to transform our R strain into the S strain. So what this did, what his experiment allowed us to do now was, which we will go over right now with Avery, McLeod, and McCarthy, was that something in this S strain allowed the R strain to be reverted back into a lethal form, and we should figure out what that exactly is. You know, if it was protein that allowed this to happen, then therefore the proteins are the cause of, proteins actually store the genetic information of our cells and stuff like that, okay? Or if it was DNA. So what these guys did, they basically did the exact same experiment where they took the S strain, they've boiled them, they've added the R strain to the same mixture, and then what they did here instead, they added a new thing that they've to confirm whether proteins or DNA or RNA or lipids that were a factor in transforming our cells. What they've added here, they added enzymes that can break down these various macromolecules, and then based on what samples you have, based on what what enzymes you added, you can sort of infer from the information that you have on which mice died and which ones ended up surviving. So what they did here, they added enzymes that can break down macromolecules. And then they've added this into the mice. And then you would look for if they would die or if they would survive. So basically that was their whole entire point. The macromolecules that the enzymes that they added were essentially lipases to break down lipids. They've added lipases, proteases, deodases, RNase, and essentially these are the ones that they've added. But basically the lipases break down lipids, the proteases break down proteins, DNases break down DNA. And then you have the RNases, which break down RNA. So basically what they did here was they've added these 4 different ones into 4 different experiments. And what happened was that all these samples that had lipases, so this, and these samples all ended up having mice that would die. But then what would happen here is that if you broke down the DNA from these bacteria, from the dead S strain, if you broke down all their DNA and stuff like that, what ended up happening was that DNA from the S strain bacteria that were boiled down did not have the ability to pass on their DNA onto the R strain, and therefore the DNA samples that you added all ended up surviving. The mice ended up surviving. So therefore DNA is the source of genetic information for these bacteria. So hopefully that made sense. And that's basically the whole gist of their experiment. What ended up happening was that, if you can already kind of observe this, was that all of these samples all have some sort of a enzyme involved, and enzymes are all protein, right? All those 4 samples are all proteins. And what some people ended up rebuting the fact was that these individuals, they've all still have some sort of a protein left in them. Even this enzyme that breaks down proteins, you still have that enzyme left in those samples. So you're not technically removing all the protein from these samples. So basically that was their whole entire thing, especially with this one, because you're seeing that the mice are surviving, you still have some protein left in that sample, which is DNase, that may be causing the mice to allow them to survive, okay? So basically that's their whole entire rebuttal. And then that caused us to go into Hershey and Chase's experiment. So their experiment basically uses bacteriophages. So bacteriophages are basically viruses that infect bacteria. And what's happening here is that they want to determine if the genetic information is found on proteins versus on DNA. So the way how they could do this is by basically adding a radioactive isotope. So they would use a radioactive isotope, either phosphorus or sulfur. And what this radioactive isotope is, it's basically something that you can use to visualize where these molecules are going. Because DNA has phosphate backbones, that radioactive phosphorus would be located on the DNA, and then radioactive sulfur will be located on proteins, because proteins use quite a bit of sulfur in them, okay? So basically their experiment was pretty simple. They took the bacteriophages plus the bacteria, and they've either added the radioactive phosphorus or the radioactive sulfur into the mix. They let the bacteriophages grow a little bit. And then they took those radioactive bacteriophages and then they've added them into a fresh batch of bacteria, right? So they took those. So this is the bacteria with the bacteriophage right here. And let's say this one has, we're going to call the DNA, we're going to call the radioactive phosphorus purple. And then the sulfur, we're going to call that as a yellow. We're going to label it. So basically what will happen here is that, during this process when you're infecting the bacteria, what you're going to do here is you're going to chop up the bacteriophage into pieces using a blender. So you're going to blenderize your media. And basically what that's going to do is going to chop up all the cells in that mixture, creating a homogenate. And then so you would create a homogenate, and then you would take that homogenate and then you would centrifuge it. Basically you would have your tube of media of your cells and stuff like that. And then all of your cells after centrifugation will be located on the bottom of your tube. And then you have your media over here, your liquid, your supernate, okay? And then that blue stuff right there is basically bacteriophages plus bacteria all blended up together. So basically their goal was to figure out, after this infection process, what are they shooting into the cells that are causing these bacteria to produce a lot of viruses, okay? Because that's what viruses do. Viruses infect a cell, they add their genetic information, and then after that genetic information, those bacterial cells end up making more bacteriophages. So they're trying to figure out what is located in that pellet compared to the supername. So what happened was that in their radioactive phosphorus group, so their P group, what happened was that if we drew the tube again, we ended up seeing all the radioactive phosphorus glowing on the bottom of the tube, whereas the sulfur group, we ended up seeing something different. We ended up seeing the sulfur located in the supername. So because of this, you can make the conclusion that the bacteriophages are inserting their genetic information as DNA into the bacteria, and then that DNA is causing the transformation of the bacteria to produce more viral particles. So basically that's their whole entire gist of this first section of chapter 6, where we essentially talk about these three different groups and how they've determined that that DNA contains the genetic information. But yeah, that's basically it. I'll see you guys later.